Hello students, welcome to EPG Pathshala. I am Dr. Amit Bhattacharya from Ramjas College, University of Delhi. Today we are going to discuss about a module, Plasmodium, Mechanism of Drug Resistance, under the paper Biology of Parasitism. After studying this module, you will be able to first know malaria incidences and status of drug resistance in India. Second, learn about reduced sensitivity to antimalarial drugs. Third, identify potential resistance molecular marker in the resistant parasite. Fourth, evaluate mechanisms for emergence of drug resistance. Fifth, analyze the urgent need to protect artemisinin combination therapies to stop malaria catastrophe. Till date, Plasmodium resistance to antimalarial drugs have been documented in 3 of the 5 malaria species known to affect human, which includes Plasmodium falciparum, Plasmodium vivax, and Plasmodium malariae. Parasite resistance results in a delayed or incomplete clearance of parasites from the patient's blood when the person is being treated with an antimalarial. The problem of antimalarial drug resistance is compounded by cross resistance, in which resistance to one drug confers resistance to other drugs that belong to the same chemical family or which have similar mode of action. During the past decades, several antimalarials had to be removed from the market after spread of malaria parasite resistance. Antimalarial drug resistance is a major public health problem which hinders the control of malaria. In India, resistance to Plasmodium falciparum to chloroquine, the cheapest and most used drug, was first reported in 1973 from Assam state. The Directorate of National Vector-Borne Disease Control Program has been monitoring the response of antimalarial drugs in Plasmodium falciparum malaria parasites in the country since 1978 through 13 monitoring teams located in the 11 regional offices for health and family welfare. Today, World Health Organization recommends artemisinin-based combination therapies for the treatment of uncomplicated malaria caused by Plasmodium falciparum. Artemisinin combination therapies have been integral to the remarkable recent successes in global malaria control, and there is broad consensus that protecting the efficacy of these drug medicine combination is an urgent priority. However, Plasmodium falciparum resistance is now emerging to artemisinin and have been detected in 5 countries, of which includes Greater Mekong sub-region, Cambodia, the Lao People Democratic Republic, Myanmar, Thailand, and Vietnam. Containment activities are ongoing in all malaria endemic countries as a part of a multi-stakeholder efforts. In 2015, it is estimated that 15 countries accounted for 80% of these cases, and these 15 countries account for 78% of malaria-related deaths. The global burden of mortality is dominated by countries in sub-Saharan Africa, with the Democratic Republic of Congo and Nigeria together accounting for more than 35% of global total of the malaria estimated deaths. Decrease in case incidences and mortality rates was slowest in these countries that had the largest number of malaria cases and deaths in 2000. Reductions in incidences needed to be greatly accelerated in these countries if global progress is to improve. The above picture shows the latest WHO malaria report, which suggests that there is a decline in malaria incidences which has fallen by 30% globally, and malaria mortality rates which has fallen by 47% globally between the year 2000 to 2013. 96 of the 97 countries with ongoing malaria transmissions have implemented a policy in their countries to test all suspected malaria patients before starting antimalarial drugs. Of these, 88 countries conduct malaria diagnostic tests free of charge in the public health clinics. In 2013, 79 of 87 malaria endemic countries adopted Artemisinin combination therapies in its national policy as the first line treatment for Plasmodium falciparum cases. The above graphical representation shows the WHO estimates between 2000 to 2015, which shows that there is a decline in malaria cases incidence rate as well as decrease in malaria mortality. So the graphical representation shows us a representation of the estimated malaria incidences rate and estimated malaria mortality rate, which is shown in blue color, while the 75% target, which is the goal of the WHO, is shown in red color. Malaria interventions have been highly effective and affordable. The main interventions comprises of vector control mechanisms, which include insecticide use, and the second one is the chemoprevention, which uses the antimalarial drug. Over a period of time, there have been some emerging challenges in front of WHO, which include insecticide resistance. The effectiveness of insecticide-based vector control method is threatened by malaria mosquito, which has developed resistance to some of the insecticide used in insecticide-treated bed nets and indoor residual spray. Since 2010, of 78 countries reporting the monitoring data, 60 countries have reported resistance to at least one insecticide in one vector population, while 49 of the 78 countries has reported resistance to insecticide from two or more classes of insecticide. Pyrethroid, one of the generally recommended insecticide in the indoor residual spray, has got resistance and have been reported from major malaria endemic countries. However, long-lasting insecticidal nets remain one of the effective measures despite resistance against the insecticide. The second major emerging problem for WHO is the reported resistance to most of the antimalarial drugs. The clinical usefulness of most of the antimalarial drugs have been compromised by the evolution, emergence and spread of resistant malarial parasite. Overuse and misuse of these antimalarial drugs are contributing to the growing resistance to the treatment for the very disease, that is burdening the low-income countries further. Resistance to the chloroquine, for example, have been reported from 81 of the 92 countries which uses chloroquine as one of the treatment regimes. And most of these countries have now shifted to second and third line antimalarial medicines for the subsequent uses. The above pictorial representation shows the distribution and report of resistance to some of the common antimalarial drugs, which includes chloroquine and sulfadoxine. We can see from this graphical representation most of the malaria endemic countries, which includes the Southeast Asia, Africa and some parts of Latin America, the chloroquine and sulfadoxine pyrimethamine resistance have been reported. Even in many of the parts of the countries which include the Southeast Asia, especially the Thai-Cambodia border, there are reports of multi-drug resistance. So all these reports are highly alarming because chloroquine and sulfadoxine pyrimethamine acts as one of the major partner drug along with artemisinin. So most of the countries where the report of resistance are coming in, they have to shift their drug regimes to some other or alternative antimalarial drugs. If we see the status of drug resistance in India, we can see that India has the world's largest population at the risk of malaria, with about 85% of the individual living in malarious countries. Chloroquine, one of the frontline drugs in the antimalarial arsenals from 1940s until the past decade, have fallen prey to the resistance, and Some of the reports in India also have reported that chloroquine resistant strains have been developed. So, the Directorate of National Vector-Borne Disease Control Program has been monitoring the response of antimalarial drugs in Plasmodium falciparum malaria parasite in the country since 1978. It is monitoring these protocols through 13 monitoring teams located at 11 regional offices of health and family welfare, with an objective to assess the therapeutic response of Plasmodium falciparum to currently used antimalarial in order to establish and generate information on sensitivity of local strains for formulation of national drug policy and recommend needful changes in the control strategy including treatment policy to contain resistant Plasmodium falciparum foci. As we know, chloroquine resistance is mainly conferred by the mutation in the gene CRT, while in the case of pyrimethamine and sulfadoxine, it is conferred by the gene mutation in DHFR and DHPS respectively. While in the case of artemisinin, Kelch domain protein, which is PF Kelch 13 mutation, has been reported. So in this tabular representation, we can see the antimalarial drug resistant gene products which are being mutated and the positions and in the consequent drug resistant alleles which are mutated. So in the case of pyrimethamine, the DHFR gene is mutated, while in the case of chloroquine, the CRT gene, which is CRT76 point mutation, is one of the major point mutation which is responsible for chloroquine resistance. While in the case of sulfadoxine, DHPS gene is responsible, while in the case of artemisinin, Kelch domain protein PF Kelch 13 mutations are responsible. So this tabular representation shows the alleles where the mutations have occurred and what are the genes which are involved in the drug resistance, which comes from the low to intermediate level or high level of drug resistance. Some of the key biochemical mechanisms of drug resistance developed by target cells or organisms to a particular drug can be: first, lesser drug delivery; second, reduced drug uptake; third, augmented drug efflux; fourth, reduced metabolic activation of the drug; fifth, improved deactivation of the drug; sixth, drug sequestration to inhibit its interference with target site; seventh, amplification of target site leading to increased intracellular concentration; eighth, target site modification; ninth, doubling of the target site activity; and 10th, improved repair of damaged target site. The diagram representation shows the basic mechanisms how a parasite attains the drug resistance. In a normal case, as the drug enters the parasite, it goes and targets certain molecules within the parasite, causing the damage to the target or the inhibition of the target. But as the parasite develops the drug resistance, there are a number of mechanisms which it develops so as to decrease the effect of this drug activity. Some of these efforts include lesser drug delivery. There is a reduced drug uptake which takes place. The drug efflux takes place at a higher rate. There is a reduced metabolic activation of this drug, improved deactivation of this drug, subsequently the amplification of the target site which causes increase in the intracellular concentration. There is a doubling of target site function and improved repair of the damaged target site if it is affected by the drug response. So all these biochemical mechanisms are the basic mechanisms through which the drug resistance to a particular drug is achieved by the microorganism. Plasmodium falciparum has developed resistance to practically all antimalarials in past and current use, even though the geographical epidemiology of resistance varies greatly to any single antimalarial drug. However, the arsenal of anti-plasmodial medicines is limited, and this is worsening by the appearance of drug resistance in parasitic protozoa. With no effective vaccines yet within reach or slow development of novel drugs, the emergence of drug-resistant parasites is becoming a major health problem in coming years. And if we see the diagram, the tabular representation over here, all the 6 WHO regions are showing significant rise in the resistance reported. So in the Central America, there is no as such report against chloroquine, pyrimethamine, sulfadoxine and methionine. While in the South America, there is a significant resistance report against chloroquine and sulfadoxine, pyrimethamine along with mefloquine, and a resistance have been reported against quinine also. In the Caribbean, there are no as such report of any resistance, while in the African subcontinent, the resistance have been reported to most of the antimalarial, which includes chloroquine, sulfadoxine, pyrimethamine, mefloquine. In the Southeast Asia, especially in India, the resistance report has been reported against chloroquine, while in the Southeast Asia and the Oceanic region, significant resistance have been reported against chloroquine, sulfadoxine, pyrimethamine, mefloquine, quinine and haloferentine. While in the East Asia, which includes China, there are significant resistance report against chloroquine, sulfadoxine, pyrimethamine, and the reports against mefloquine are still not clear. So clearly showing that there is an emergence of drug-resistant Plasmodium falciparum malaria cases significantly in some of the areas which are malaria endemic. Chloroquine-resistant strains are associated with mutations of the PFCRT genes. Possible mechanisms suggest an energy-dependent efflux of pre-accumulated chloroquine from parasite vacuole by an unidentified transmembrane protein pump, or due to increase in vacuolar pH leading to reduced drug concentration and accumulation at the target site. The PFCRT gene, in which multiple mutations are responsible for chloroquine resistance, have been mapped in a single segment of chromosome 7 of Plasmodium falciparum laboratory strains. Mutations of the positive charge amino acid lysine 76 is critical for the emergence. Evidences suggest 8-point mutations in PFCRT, which are M741I, N75E, K76T, A220S, Q271E, N326S, I356T, and R371I are responsible for chloroquine resistance, which have been distinguished from the chloroquine sensitive strains. The gene product PFCRT is a possible transmembrane transporter of the parasite's digestive vacuole membrane and may be functioning in drug flux, which includes efflux and influx, and or pH regulation. Reports from several malaria endemic regions such as Brazil, Uganda, Laos, Thailand and Papua New Guinea strongly associate PFCRT K76T mutation and chloroquine resistance in Plasmodium falciparum strains. The mechanism of resistance for chloroquine has been shown in this pictorial representation. It has been discovered that chloroquine stalls the novel heme polymerase enzyme, a vital detoxification enzyme present in the intraerythrocytic food vacuole. Chloroquine cannot be effluxed out from the digestive vacuole of chloroquine-sensitive parasites due to the presence of PFCRT lysine 76 amino acid residue, leading to formation of lethal chloroquine heme complex. While in the case of chloroquine-sensitive parasite which lacks lysine 76 residue and contains other point mutations, chloroquine is expelled via mutated PFCRT by energy-dependent efflux mechanisms. The geographic distribution of chloroquine-resistant and chloroquine-sensitive strains are shown in this image. If we see, the resistance to antimalarial drugs such as chloroquine is widespread throughout much of Africa and other parts of the developing world, which include Southeast Asia and India, where malaria transmission is on rise and are highly transmitted in these areas. The problem of chloroquine resistance started from Thai-Cambodia border in the year 1961, which further moved to Bangladesh, northeastern states, Odisha, Madhya Pradesh, and subsequently spread worldwide from at least 6 known origins. So if we see the image over here, the spread of chloroquine resistance is shown over here, which started from the Thai-Cambodia border in the 1960s and subsequently moved to the other part of the world. The map above shows the chloroquine resistant areas in Indian subcontinent. Chloroquine resistant Plasmodium falciparum cases have been reported from number of districts of high importance. Of these, 117 high endemic districts are from 7 northeastern states: Andhra Pradesh, Chhattisgarh, Jharkhand, Madhya Pradesh and Odisha. So these states contribute to number and majority of the chloroquine resistant cases which have been reported across the country. Slide 25. Chloroquine and sulfadoxine pyrimethamine, which is marketed as Fansidar, have been the two low-cost and most widely used antimalarial drugs, but widespread resistance to them has turned out to be a decisive global community health challenge. Sulfadoxine pyrimethamine is an antifolate synergistic drug combination that block two vital enzymes of parasites folate biosynthesis pathway through sequential and synergistic inhibitory mechanism. Pyrimethamine blocks the activity of dihydrofolate reductase enzyme, while the sulfonamides block the activity of dihydroterioate synthase enzyme. Plasmodium has the capacity to exploit host plasma folate as its primary folate source. Folate is a broad term that represents 9 folate derivatives, which include folic acid, that are found in mammalian and microorganism cells. Folates, an essential cofactor, are required for both malarial parasite development and production of host erythrocytes. They are involved in delivering one carbon unit to 3 major metabolic pathways, which are biosynthesis of amino acid methionine, purines and pyrimidines. While the biosynthesis process of purines and pyrimidines are crucial for DNA synthesis, thus the inhibition of enzymes involved in these pathways greatly affect the activity of the dividing cells and stop the DNA and protein synthesis. Interruption of folate biosynthesis pathway by DHFR and DHPase inhibitors creates reduced level of completely reduced tetrahydrofolate, a crucial cofactor required during one carbon unit transfer in the purine and pyrimidine and amino acid methionine biosynthesis pathway. Sulfadoxine pyrimethamine combination resistance have been linked to genetic mutation in 2 genes, the dihydrofolate reductase DHFR gene and dihydroterate synthase DHPase gene, and for half-life of the drug combination. In addition, clinical treatment failures also befalls as the parasite develops adequate resistant mutation to overcome therapeutic doses of sulfadoxine pyrimethamine drug combination. Multiple mutations in PF-DHFR gene and double mutation in DHPase gene that collectively form the quadruple mutations are associated with sulfadoxine pyrimethamine resistance. The rapid spread of sulfadoxine pyrimethamine resistance had led to malaria endemic country to eventually replace sulfadoxine pyrimethamine with the currently available Artemisinin based combination therapies. The above picture shows the widespread distribution of resistance against sulfadoxine pyrimethamine drug. So we can see from the image that the sulfadoxine pyrimethamine resistance have been spread out to most of the part of Southeast Asia, Africa and Latin America or South American regions. One of the main important factors of this widespread increase is repeated and unadministered use of these chemicals. The extensive deployment of these conventional antimalarials in the past 50 years have caused tremendous drug selection pressure on Plasmodial parasite, which has developed mechanisms to resist these drugs. The above picture shows how the different conventional antimalarial drug, which includes chloroquine, sulfadoxine, pyrimethamine, quinine and mefloquine, has a declining response. All these reasons are due to the unadministered and extensive use of these drugs. So we can see from this image at the various interval of time they have fallen prey to the drug resistance. So we can see the first drug which got resistance against it was chloroquine. Subsequently sulfadoxine pyrimethamine resistance was reported. Then mefloquine and quinine came into the picture. So it's very essential to stop the development of resistance against these drugs at the very early level. Artemisinin, isolated from the plant Artemisia annua, is the cornerstone of anti-plasmodial drugs. Artemisinin are the most powerful medicines currently available to treat uncomplicated to severe Plasmodium falciparum cases because of its ability to swiftly reduce the intraerythrocytic parasites in the patient with malaria. Artemisinin is an sesquiterpene lactone isolated from the shrub Artemisia annua and has been reported by Chinese herbalists as medicine for more than 1000 years in the treatment of skin diseases and malaria. The earliest known documentation of artemisinin was in the book 52 Prescriptions revealed in the Han Dynasty in 168 BC. Malaria was traditionally treated by chloroquine or quinine, but with declining success. By the late 1960s, efforts to eradicate malaria had failed and the disease was on rise. At that time, Professor Yo-Yo Tu in China turned to traditional herbal medicine to tackle the challenge of developing novel malarial therapies. From a large-scale screening of herbal remedies in malaria-infected animals, an extract from the plant Artemisia annua emerged as an interesting candidate. Now, artemisinin represents a new class of antimalarial agent that rapidly kills the malarial parasite at an early stage of their development, which explains its potent and uprising activity against malarial parasite. Emergence and spread of resistance to artemisinin elevates the danger of wiping out present decline in malaria burden worldwide, as the efficacy of currently recommended artemisinin combination therapies as first line of treatment in chloroquine and sulfadoxine resistant areas will be compromised. The main reasons of the development of this artemisinin resistance is because over the years, due to prescription of wrong combinations, wrong dosages and extensive use of oral artemisinin based monotherapies has resulted in emergence of some resistant parasites, making the artemisinin combination therapy medicines useless and ineffective. The researchers' teams, which include collaboration between Mahidol University, Thailand, Oxford University and the Wellcome Trust, conducted the DNA sequences of the 940 Plasmodium falciparum malaria parasite samples which were collected between 2013 to 2014 from Myanmar and adjoining border regions of Thailand and Bangladesh. Of these 940 collected samples, which are the malaria infected samples, 371 samples, which is about 39% of the samples demonstrated artemisinin resistant marker, which is Kelch K13 propeller mutation. Recently, in 2015, the first genotyping studies were conducted in India to locate PFK13 point mutations in Plasmodium falciparum samples. Now, the K13 propeller polymorphism is regarded as a handy molecular marker for surveillance effort to trace artemisinin-resistant parasites across the malaria endemic regions. In a recent update, artemisinin resistance is established in Myanmar and confirmed artemisinin-resistant parasites have emerged in Homelin, which is located within 25 km from Indian subcontinent. The above image shows the representation of homology between Plasmodium falciparum K13 and human KEAP1 protein. The structural 3D molecule model of K13 propeller domain is also shown for the comparison. The loss of artemisinin derivatives or artemisinin combination therapy would increase the malaria burden drastically, leading to severe consequences on public health in malaria endemic nations. The catastrophe would be devastating as no alternative antimalarial drugs are ready for commercial use in next few years so as to replace artemisinin combination therapies. Scientists in 2008 reported the first cases of artemisinin-resistant Plasmodium falciparum in West Cambodia. In January 2014, the reports on artemisinin-resistant published by WHO Global Malaria Program documented that the epicenter of artemisinin resistance have been recognized in 5 countries in the Greater Mekong sub-region: Cambodia, Lao People Democratic Republic, Myanmar, Thailand, and Vietnam. Additional foci is suspected in Suriname, Guyana, and French Guiana, France. Today, one of the foremost concerns of World Health Organization is that artemisinin derivatives resistance may widen beyond the Greater Mekong sub-region and independently emerge in malarious region of other continents. In the image above, we can see that the areas which are in that region have been spotted red in color, which shows that the artemisinin resistant strains have been confirmed from these particular regions. These include some parts of India, Bangladesh, Myanmar, Thailand, Laos, Cambodia, Vietnam. The emergence of Plasmodium falciparum resistance to artemisinin is an urgent public health concern, due to which WHO in the year January 2011 released the Global Plan for Artemisinin Resistance Containment, which put forward 4 main goals and recommendations. First, to stop the spread of resistant parasite. Second, to increase monitoring and surveillance to evaluate the artemisinin resistance threat. Third, to improve access to diagnostics and rational treatment with artemisinin combination therapies. And fourth, to invest in artemisinin resistance related research. Southeast Asia region has reported the antimalarial drug resistance to most of the conventional antimalarials, which includes chloroquine, sulfadoxine, mefloquine, and latest is the artemisinin. Incomplete coverage of malaria-affected population, inadequate treatment, and unwarranted employment of monotherapy are the main grounds of emergence of drug resistance in this area. Further, poor quality and substandard drugs in the market are another main reason why the tropical countries are developing the resistance against most of the antimalarial drugs. So students, let us summarize what we have learned in this module. More than 3.4 billion citizens are at a risk of contracting malaria, and each year it claims more than 4,50,000 lives predominantly among children and pregnant women. Plasmodium falciparum, the deadliest form among the 5 malaria parasite species, is accountable for the greater part of malaria-related mortality and morbidity. Artemisinin combination therapies are presently the most effective and frontline drug against Plasmodium falciparum malaria. Due to extensive and excessive use of artemisinin monotherapy, there is a serious public health concern that parasites would attain widespread resistance against this critical treatment. Plasmodium falciparum in most malaria-affected areas is highly resistant to chloroquine, one of the earliest drugs discovered, due to its overuse, while the resistance to sulfadoxine pyrimethamine is also widespread. The mefloquine resistance has been confined from some regions where it is being used extensively, such as Thailand, Cambodia, and Vietnam. Chloroquine resistance is primarily caused by mutation in a gene PFCRT that encodes a transmembrane transporter PFCRT, while the atovaquone and pyrimethamine resistance is conferred due to a single point mutation in the gene encoding cytochrome B and dihydrofolate reductase respectively. Artemisinin and its derivative is an essential constituent of treatment regimes from multi-drug resistant falciparum malaria, and recently few areas in Southeast Asia regions have reported artemisinin resistant cases. Incomplete coverage of malaria affected populations, inadequate treatment, and unwarranted employment of monotherapies are the main grounds of emergence of drug resistance. Further, poor quality drugs in the market, which are common in tropical countries, is often regarded as a major concern. The steady appearance of Plasmodium falciparum resistance to many antimalarial medicines over the past several decades has since led malaria endemic countries to adopt artemisinin-based combination therapies. Protecting the efficacy of artemisinin-based combination therapies as the current first-line treatment for Plasmodium falciparum malaria is now among the top global public health priorities. So with this note, I conclude my lecture. Thank you very much.